What? Oh, I'm just gonna leave this like this, and we're gonna start talking. Okay. Hopefully the internet doesn't keep going out. Okay, so today we're gonna be discussing the exam one. So first off, microbes require a microscope to be seen, and it's from millimeters to zero point 0.2 micrometers. Exceptions are supersized cells, microbial communities, and viruses. Early scientific difficulty because of the microscopes were too low resolution, and species of microbes were hard to define. The electron microscope by Ernst Ruska and ultracentrifuge by Theodor Svedberg helped microbe research. Protista are organisms that do not fit into the kingdom of plants, animals, or fungi. The 16S rRNA gene differentiated the archaea from bacteria within the domain Monera. Archaea also have ether-linked lipids and no peptidoglycan, whereas bacteria have ester-linked peptidoglycan cell walls. The Robert Whittaker kingdom is about protists, which are protozoans and algae, and minerals, which are bacteria. Or sorry, what? What am I saying? Protists, Monera, fungi, plants, animals. Endosymbiotic theory is by Lynn Margulis. This is basically that mitochondria and chloroplasts evolved from prokaryotic cells. The evidence is DNA, ribosomes, and morphology. So we're going to talk about some people. Robert Hooke developed the first compound microscope and coined the term cell. Anton van Leeuwenhoek developed the single lens magnifiers to observe microbes. Francisco Redi proved that maggots and decaying meat were offspring of flies. John Needham and Lazaro Spallanzan was a sealed flask of meat didn't grow microbes. Mr. Onesimus and Lady Montague introduced smallpox inoculation in America and Europe. Edward Jenner deliberately infected patients with cowpox matter. Florence Nightingale recognized disease warfare and devised the polar area chart. Louis Pasteur developed the Sonnec flask basis of fermentation and disproved spontaneous generation and proposed biogenesis. He developed the first vaccines based on weakened cholera and rabies. Robert Koch determined the germ theory, scientific method, and chain of infection from anthrax. So he has basically three, but four postulates. The first one is a microbe is always present in disease host and absent in healthy. 2, microbe is grown in pure culture with no other microbes present. 3, introduce pure microbes into healthy hosts and the individual will become sick. And then 4, the bacteria is derived from sick or affected individual. Ignaz Semmelweis ordered doctors to wash their hands with antiseptic chlorine. Joseph Lister developed carbolic acid to treat wounds, asepsis for surgery. Alexander Fleming rediscovered penicillin. Sergei Winogradsky basically just studied environmental microbiology, basically. And then Carolus Linnaeus, he is the father of modern taxonomy and helped name everything. Lynn Margulis is the endosymbiotic theory. Carl Woese discovered archaea, a distinct prokaryote with DNA that codes for ssRNA. Frederick Griffith discovered the transformation in bacteria. Oswald Avery, transforming substances DNA. Rosalind Franklin, double helix shape of DNA. Watson Crick, the basis and anti-parallel nature. Listen up. The refractive index. For vacuum or air, it's 1. For water, it's 1.3. And then for glass, it's 1.5. In 10 to the power of negative 3 increments. The biggest is the meter. Then a millimeter, which is 1,000th less. than the meter. And then the micrometer, which is the 1,000 less than the millimeter. So basically, it goes meter, millimeter, micrometer, nanometer, and then picometer. Microscopes. So you have light microscopy. This includes bright field, dark field, and phase contrast. So bright field uses short wavelength light, increase contrast and aperture, and then immersion oil enhanced by fixation, which is adhesion, and then staining, and also wet mount prep. It can resolve up to 200 nanometers. Dark field detects unresolved objects, resolves thin and live microbes. Phase contrast used to view live cells and cellular organelles. Um, Moving on to fluorescence microscopy, the specimen with the fluorophore obtains shorter wavelength light, which is basically excitation, and then fluoresces a beautiful, colorful, longer wavelength light, which is emission. Immunofluorescence is when a fluorophore is combined with antibodies against a specific bacterium. For example, the VRDL syphilis test for Treponema palladium. Electron microscopy. This uses beams of electrons instead of light rays. So TEM is transmission electron microscopy. It uses a slice of cells up to 100,000 times and looks at internal 2D structure and goes from nanometers to micrometers. SEM is scanning electron microscopy. This is metal coated cells. It resolves up to 10,000 times and looks at external structures, 3D. And this resolves from a micrometer to a millimeter. Okay, the numerical aperture allows for greater resolution. So numerical aperture equals n multiplied by sine theta. So n is the refractive index. So like speed over volume. Again, 1 for air, 1.3 for water, 1.5 for glass. Resolution is the smallest distance two objects can be separated and still be distinguished. A greater resolution is a greater numerical aperture and lower wavelength. Detection is ability to determine presence of an object. Magnification or refraction is a bending of light as it enters a substance that slows its speed. Bacterial shapes, you got cocci, bacilli, and spiral. So basically, spirochetes have a flexible cell wall, and spirola has rigid. Arrangement, you have staph, strep, di, and tetrad. Stains, you have simple, which adds color to cells but not external medium. For example, methylene blue. And you have differential, which stains one type of cells. So basically, gram stain and acid fasting. How a gram stain works is you kind of have water, then you have a methanol fix, then you have the primary stain, which is crystal violet, and then you have the mordant, which is iodine, and then you have the decolorizer, which is the ethanol, and then you have the counter stain, which is safranin. Going back, so staph means clusters, strep means chains, di means pairs, and tetrad means a square. Okay, so prokaryotic structure, you got the cell membrane with phospholipid proteins and no sterols except for mycobacteria. And then the cell wall has peptidoglycan, teichoic acids for positive bacteria, and liposaccharide for negative bacteria, and then mycobacteria have no cell wall. Mycobacteria, we'll talk about them uniquely. So they have cell envelopes made of mycolic acids, which are lipids, and then arabinogalactans, which is sugar, and they can be only stained with acid fasting, thrive by choosing isotonic environments. Bacteria and archaea, so basically their similarities, they have the 70S ribosome, circular DNA, phospholipid bilayer, and no nucleus. So bacteria have peptidoglycan, ester-linked acids, simple RNA, pathogenic, and are aerobes/anaerobes, whereas archaea have pseudopeptidoglycan, ether-linked isoprenoids, complex RNA pol, and extremophiles. Active transport moves against gradient, and then passive moves down. Gram-bacteria, so positive has thick peptidoglycan. It holds a purple stain, has teichoic acids and no porins or outer membrane. Negatives have thin peptidoglycan, holds pink stain, outer membrane, lipopolysaccharide, and have porins. Positive examples are Staphylococcus, Enterococcus, and Streptococcus, and negative examples are E. coli. A capsule is made of polysaccharide and glycoprotein and protect from phagocytosis, and then the S layer is made of protein or glycoprotein and protect from osmotic stress. Okay, now that we're done with that, I'm gonna move on to the structure of the cell. Okay, so basically, each basically each gram bacteria has a chain of a glycan chain. It is NAG and NAM. So NAG is N-acetylglucosamine, and then NAM is uh, NAM is N-acetylmuramic acid. Okay, so all of them have this. So it'll be NAG, NAM, NAG, NAM, NAG, NAM. Basically, that's the uh, kind of like part of the peptidoglycan backbone. Okay, so from that second NAM, you go down. There's a whole bunch of amino acids called the tetrapeptide chain. That's 4 of them. So first you got L-alanine. Second, you got D-glutamic acid. Third, you have either diaminopimelic acids, also known as I think DAP. That's for negative bacteria, whereas L-lysine, I think, is for gram-positive bacteria, and then the fourth one will be D-alanine. So again, L-alanine, D-glutamic acid, and then for negative it's DIP, and then for positive it's LYS, and then fourth is D-alanine. So starting from the DIP slash LYS, gram positive will have the peptide interbridge. Gram negatives also have a peptide bond, but it's not like an interbridge. This one has a pentoglycine interbridge for gram positive. So yeah. And then so basically from that basically from that third amino acid, it'll connect to the first amino acid on another chain. So it'll start with D-alanine, then go to DIP slash LLYS, then to D-glutamic acid, then to L-alanine, all the way down to NAM again, like NAM, and then NAM, NAG, NAM, NAG, NAM, NAG. Basically, it goes kind of on and on like that. So those are the difference between that. So with gram-negative bacteria, LPS is pretty important. So it kind of has the outer membrane and then the inner membrane with the peptidoglycan in the middle between those two membranes, right? So the outer membrane has the LPS on it. The LPS is pretty important. It has the glucosamine phosphate dimer as like kind of like the zigzaggy middle, and then the fatty acid tails are on the bottom, and then on the top you got the core polysaccharide that connects to the O polysaccharide, and the O polysaccharide is really important. Um, reading kind of like down, you have like the man section, Abe section, THA section, gal section, then the gin section. Don't know what that has to do with anything, but then the fatty acids have oxygen and ammonia, and then the whole bottom part is called lipid A, and the top part is called the polysaccharide part, core polysaccharide, O polysaccharide. Okay, that should be it. I will repeat the structure for you again. It goes, the backbone goes NAG, NAM, NAG, NAM. From the second NAM, it goes L-alanine, D-glutamic acid, then the DIP or LYS. DIP is for negative, LYS is for positive. Then it goes D-alanine, right? Okay, so from the DIP slash LLYS, you have the peptide interbridge. Then it goes D-alanine, DIP slash LLYS, D-glutamic acid, L-alanine, it's in there. Um, I'm gonna search something up really quick and then I'm gonna end this. It is very loud and cold. Okay, I guess we'll never know. Okay, so in gram-negative bacteria, there are no pentaglycine interbridge. Instead, gram-negative bacteria have direct cross-links between their peptidoglycan strand. So they have the cross-link between VIP and B-ALA. So it's like a direct cross-link. So for gram-positive, you have the pentaglycine interbridge. Okay, that's it. Goodbye. Good luck on the test.